when you talk about muscle function, we're talking about contraction and relaxation. Muscles have this great property of lengthening and shortening. And when they contract, they shorten. And when they do that, they move things. So it's very important to know it, because without it, we wouldn't be able to move. Birds wouldn't be able to fly. Fish wouldn't be able to swim. Animals, humans, we wouldn't be able to run, walk, jump. So it's important to know. And grossly, the muscle has a belly attached to the bones via tendons, and within the substance of the muscle are fascicles, and those fascicles hold the muscle fibers. So muscle fibers are actually the name given to muscle cells. The muscle cells actually fuse to form the muscle fiber, and that's why the muscle fiber has numerous nuclei. It's multinucleated, and within the muscle cell are these myofibrils, which are like rod-like structures which arrange longitudinally, and they contain the functional units of the muscle, which is the sarcomere. So the sarcomere is basically an arrangement of thick and thin filaments, and it's thick filaments called myosin and thin filaments which consist of four parts, two which are structural and two which are regulatory. The two structural filaments of the thin filament are the actin and the nebulin. Actin is the basic subunit of the thin filament, which interacts with the myosin during the excitation, contraction, coupling, or the sliding filament process. And the nebulin is basically a scaffold that holds the actin together, and you have like a intertwining arrangement of the polymeric subunits of actin, and around it is the regulatory filament, which is tropomyosin, and the tropomyosin is a filament that covers the active sites where myosin attaches, and attached to the tropomyosin is troponin. Troponin has three parts: troponin T, troponin I, and troponin C. Troponin T binds to tropomyosin, troponin I inhibits the actin, and troponin C is where calcium binds. So what happens is that this tropomyosin conceals the active site where the myosin attaches to form contractions, and attached to the tropomyosin is the troponin. And when calcium attaches to troponin C, it actually pulls the filament away from the active site on actin, enabling the myosin and actin to interact and cause contraction. So that's what happens with the thin filaments. And when you look at the thick filaments, you have myosin. And with myosin, it's basically a substance with 4 light chains and 2 heavy chains. The heavy chains make the bulk of it, and it has a tail and a head, and they intertwine and then form a head which is like a globular structure. And on that head there's actually a site where ATP attaches, and there's a site which attaches to the actin. And when you look at the sarcomere, the myosin is in the center, and the actin is peripherally. Myosin is linked to an M strip in the center of the sarcomere, and then it forms a mirror image on the other side. And the myosin is held to the Z line via a scaffold kind of protein known as titin, and the titin holds it in place. And then from the Z line extending towards the middle are your actin filaments, or your light chain, or your light filaments. And these actin filaments interdigitate with the myosin filaments. And when you look at this microscopically, you have dark bands and light bands, and the dark bands are where the thick and thin filaments interdigitate. When you have only actin, you have an I band, which is light. Where they interdigitate, you have an A band, which is dark. And then when you only have myosin, you have an H band, which is light. And what happens with excitation, contraction, coupling, is that the amount of overlap increases, so the A band actually lengthens. The whole sarcomere shortens, but the amount of overlap increases, so the A band increases in length. So this whole process occurs when an action potential arrives at the neuromuscular junction, and you have the end plate potential, and then the action potential moves in two directions of the muscle cell, from one side to the next, and then within the muscle cell are holes, and these holes penetrate right through the muscle cell, and they're called T-tubules, and flanking the T-tubules are sarcoplasmic reticulum, and this sarcoplasmic reticulum contains calcium. And what happens is that when the action potential descends down these T-tubules, they stimulate a receptor known as a dihydropyridine receptor, and that stimulates the ryanodine receptor, which is on the sarcoplasmic reticulum, and that causes the release of calcium. Calcium then goes into the cytosol, and it binds to the C component of troponin, and that pulls on the tropomyosin, exposing the active site for actin and myosin to interact and cause contraction. So what happens is that the myosin, which is now charged up from dephosphorylation of ATP, then latches onto the active site and moves into position and causes the power stroke, which results in contraction. And with that, the muscle shortens. The A band increases, meaning the overlap increases. But the thing is, you need to detach the myosin head from the actin to enable another contraction to occur. And there's two myosin heads, so they happen in step. And ATP is needed to detach the myosin head from the actin. And when it does that, it then dephosphorylates to charge up the head so that it's sprung into position so that it can act again by attaching to another active site on actin. So ATP is involved in charging up the myosin head into that position where it can spring and cause that power stroke. And it's also involved in detaching the myosin from the actin and causing uptake of calcium at the sarcoplasmic reticulum. So ATP is actually mainly involved in the relaxation process. Calcium is involved in the contraction process because calcium exposes the active site, facilitating the interaction between myosin and actin, and ATP charges up the myosin head, detaches it from the actin, and causes uptake of calcium into the sarcoplasmic reticulum. So relaxation is actually facilitated by ATP, and contraction is facilitated by calcium. So that's how it functions. And with smooth muscle, it's different because the calcium comes from extracellular space, whereas in skeletal muscle, it comes intracellular from the sarcoplasmic reticulum via the linkage that occurs between the dihydropyridine receptor on the cell membrane and the ryanodine receptor on the sarcoplasmic reticulum. Okay, thanks a lot. I hope that makes sense, and keep well, and we'll talk about smooth muscle in another installation.